Hello,
Hi, good morning.
good morning.
And you're gonna do the YouTube as well, Glennis?
Yeah, it's here live already.
Great. Good morning, everybody. Welcome again to our Monty Hart Cath conference. This morning, we wanted to talk a little bit about drug-coated balloons. There's just so much happening in this field, numerous clinical studies, and I think I've mentioned to most of you, this is probably one of my favorite devices, and a device I believe is very important to have in the cath lab, and will become an important part of what we do as interventional cardiologists in the future. We've given some of this talk, me and Pierre, in many different settings in very large conferences around the world. And I thought it was time we kind of went through this with you guys too. And also, we want this to be very interactive. We really want to give you an update, but we want to answer all your drug-coated balloon questions, or as many as we can today. So at any stage, raise your hand. Please stop us. Everybody today has the ability to talk or come off mute and ask a question or ask for clarification or make a comment. So let's try and make this as interactive as possible. So me and Pierre put these slides together, and we really want to go from the basics to then the technique, data and technique, right, Pierre? So let's go to the next slide. Let's see what's...
Yes, absolutely. Thank you, Dr. Atib. Yeah, please, anyone, feel free to interrupt me or anything. And I think this is really an exciting time for DCBs. The reason why we're here today, this is just a screenshot from PubMed from a couple days ago. And you can really see that if you look up drug-eluting stent, there's been so much literature on it in past 20 years, it's been up. But you can also appreciate if you write down drug-coated balloon, well, in the past 10 years, there's really some increase that is pretty relevant in terms of the evidence that's coming up on these devices. So I think it's a pretty contemporary matter, and I'm really, really happy that we can be here with Dr. Tip today. So why DCB? Why drug-coated balloons? Well, DES, drug-eluting stents, are great. Drug-eluting stents do the job. They're very safe devices. Whenever we prepare a lesion, we're very sure that by putting a stent, most of the times we really can get a hand and get through the procedure. But in terms of long-term outcomes, on the right-hand side of the picture, this is a milestone paper from Madhavan 2020. It really shows that irrespective of the type of drug, either drug-eluting stent or bare metal stent that you implant, year by year, you accrue some events, which can be target lesion revascularization, meaning that the patient comes back for some more PCI, or patients can present with myocardial infarction or even worse. And you can really appreciate that it really does not stop, and it just tends to increase year by year.
I
And
think,
this,
if you
yeah,
look at any, and we've looked at so many studies like this is one, but there's studies now even looking at Zines up to 10 years. And no matter what we do, and this is irrespective of lipids, controlling blood pressure, doing everything you possibly can, there's this continued accrual of events that happen related to a stented artery that continue to happen with time. Some of it's TLR, but some of it also can be, severe restenosis that results in acute coronary syndrome or an MI. But, this on the right here, that graph of that continuous 2 to 3%, some studies show as high as 4%, but I think it's 2 to 3% accrual of events every year is kind of what, for us as interventional cardiologists, has been, I think, the holy grail of how do we truly compete with a lima, right? Because if I had to plot a lima graph, okay, not a vein graph, but a lima next to that, it's just going to be flat. And it's going to be flat for like pretty much 20 years, right? And someone on this call might say, yes, but it has to be a well-placed lima. Of course, technically it has to be well done. But if it is well done, it just flattens that increase curve, right? So how do we as interventional cardiologists get to a point or a technology where we can maybe, I don't know if we can ever be as good as a Lima, all right? But can we get to a point where we start to flatten this curve?
Yes, absolutely. And Dr. Latib, you've always shown also this slide with a heart with 67 stents. And this is not contemporary PCI. This is something that, happened, unfortunately, stent by stent. But I think in 2025, we should offer patients more. And whenever we go ahead and do studies and we compare drug-eluting stents with drug-coated balloons, we need to remember that these are different technologies. One is a balloon and one is a balloon with a stent on top of it. And
I would just go
stent,
back just one more second. Just I would say
yes,
one more thing about this. I really do think I remember when I started doing PCI, and I don't know, Michael, you can remember, you worked with some of the people that I worked with. You know, there was a time in the Cath lab it felt good saying, "Ah, I did a full metal jacket on this case." It was so tough. But honestly, I think the terminology full metal jacket should become historical. It should be something that goes into the history books and say, oh shit, there was that bad time when we really didn't know what we were doing. And we would put full metal jackets into people's arteries. To me, I think modern interventional cardiology, if you in a case where you're considering doing a full metal jacket, which means like to me, literally putting more than 60 millimeters of stent in, you should stop yourself and wonder, because I do think that there are very few situations nowadays where we should be doing full metal jackets. And it should be almost a term that is a historical term that we can look back and say, oh, remember on the bad old days when we had shitty, crappy stents, we used to do this thing called full metal jackets, but nowadays we don't. I honestly almost believe that it's better not putting stents in to a patient's artery and treating it medically than having to do it with 80, 100, 120 millimeters of stent in one vessel.
yes, absolutely. And we still need to remember that, right now we're living on what we did in the past year. So 10% of the PCIs today are still for treating ISR. And we have bare metal, drug-eluting stent, ISR. Of course, the relative share of this has changed a little bit over the years, but 1 out of 10 PCIs are due to ISR. But why is this important? Well, it's important because on the left side, you'd see ISR number one. So in stent restenosis number one, a procedure for ISR has more risks of target vessel revascularization, which means that the patient comes back because he has, again, pain, he has shortness of breath, he has myocardial infarction when compared to a PCI in a de novo lesion. So a new PCI not within a stent. And then on the right-hand side, we have ISR number two and number three. These are having so many events patients with recurrent ISR that is really remarkable. 1 out of 2 patients over 3 years, if they have a multi-layer drug-eluting stent ISR, they will come back. And I think we always talk about lifetime management of patients with aortic stenosis. In some way, I think it should be very, very similar also a patient with coronary artery disease. And so what are DCBs? Well, these are some technologies that allow us to do the job and leave nothing behind. And there's 4 main components: the catheter, the balloon, the excipient, and the drug. We're going to go through them with Dr. T just in a second, but I'm just sharing with you some images. Dr. T was saying more than 60 millimeter stents is not really contemporary PCI. And I think on the right hand side, you can see over here that it would be very difficult to put less than 60 millimeter stents in this artery. And I think so really over the years, we have developed so many technologies to go ahead and mitigate the risk of any other precedent technology. So we started off with POBA, so plain old balloon angioplasty. There was some risk of acute occlusion, recoil, and thrombosis, which were mitigated with bare metal stents. But unfortunately, neo-intimal hyperplasia was there. So drug-eluting stents were then developed to mitigate the risk of neo-intimal hyperplasia. Nonetheless, drug-eluting stents themselves at the risk of very late thrombosis. And BVS, so bioresorbable vascular scaffolds, they were developed to actually mitigate this, but they didn't really manage to. And they actually were associated with a higher risk of very late thrombosis. Nonetheless, they were the first devices that were made with the concept of giving back to the artery vasomotion and the possibility to remodel, because there would be no cage there that would allow eventually also late luminal enlargement. And I think with drug-coated balloons, we come back to this and we try and make this happen. of course, with the idea and the possibility of acute occlusion coming back and acute recoil that we really need to be aware of whenever we do a drug-coated balloon angioplasty. So I think in terms of what the procedure is going to look like, and we're going to go into details with Dr. Tim in just a second, I think procedure-wise, it's very important to know how to do angioplasty and kind of come back to the times of POBA. This is just a slide to show the positive vessel remodeling and late lumen enlargement. On the right-hand side, you can see that with time, the vessel can get larger. And even if maybe there's some plaque coming back, the glial effect can't really take place, and the vessel gets larger, and in the end, maybe the lumen stays the same. So we're no caging, and really starting off with a stent already gives us a baseline that is never going to increase. So the areas are going to be that, and if there's any kind of neo-intimal obturation, we're going to decrease the area. With the glial effect that you can really get with drug-coated balloon, well, then you can really sometimes also gain some lumen in the long run. I think this is very, very interesting. And we're talking about why DCBs, but how much DCBs are used all over the world. So recently, in March of last year, we had the approval of the first DCB paclitaxel for ISR in the US. But other parts of the world, they'll be using a lot. And you can look in Japan, one PCI every 5. So the ratio is 1 to 4 is DCB versus regular stent. So they're really, really active in terms. And European centers, very many European centers are really high volume DCBs. And I think it's technology that we should explore. And when it was explored in the beginning, there were some questions because of some studies that were conducted in patients with peripheral artery disease undergoing drug-coated balloon angioplasty. And some meta-analysis that shown that there might have been some safety concerns with paclitaxel-coated balloons. Well, numerous meta-analysis in different districts of the body, peripheral artery disease, but also coronary artery disease, were done to ensure that this was not the case. And this is a meta-analysis with Dr. Scheller, Dr. Tieb, and a whole team of experts that really show that drug-coated balloon angioplasty, when compared to other modalities of PCI, is not associated to a survival disadvantage.
Now, I would just stop you a little bit, on this slide. I mean, we did this paper, this meta-analysis. I'm a co-author, and we did it really at the height of all the controversy that was happening in the United States, I would say, more than the rest of the world, about the concerns of the safety of drug-coated balloons in the periphery, right? When that data came out, there was a meta-analysis showing maybe that drug-coated balloons increase mortality in the periphery. And I do believe there may be not some truth to it, but I am concerned about certain drug-coated balloons in the periphery and some of the challenges they may have. And we can talk about that if someone wants to ask the question. So we want you to really reassure our colleagues about the safety of drug-coated balloons. So we took all the randomized data that was out there, and one was a study I did, look and try to put it together to say, let's show you in a large group of patients, these were 4,500 patients, is there even a signal that drug-coated balloons in the coronaries may impact patients negatively, whether there be death, cardiac death, MI or TLR? And you see, like, nothing is going to the right of that line, right? Everything is going to the left of that line, and even things like cardiac death and myocardial infarction, maybe we found a benefit. And you'll see that that's been repeatedly when we do meta-analyses, particularly in de novo disease, we see this, right? And restenosis may not be as evident, but certainly in de novo disease. Sorry, was there a question? Michael, did you have a question? Maybe
No,
not.
I'm good.
Okay. So I think this data, if someone ever asks you, are you concerned about the safety of drug-coated balloons? I think this study kind of helps you. And the study you should know about. We published it in 2020 in JACC. We hopefully will update and do other meta-analyses like this in the future, but we'll show you another one we just recently did in small vessels.
Yes, absolutely. So we're going to get to that in just a moment. And I'm just going to focus over here on a few details. And please, Dr. T, just stop me whenever you think, we've dealt with the major issues. But I think one of the main question is, what are drug-coated balloons made of? So I think we need to realize what we're talking about.
So maybe let me do this part, Pierre, because I wanted to be a little technical on this. So this is a list of all the drug-coated balloons we know of. Okay, there's probably another 20 I'm sure we don't know about, but these are the ones we know about. Majority of these are approved in Europe and the Middle East. There are a few that are either been taken off the market or are still in clinical study and don't have approval anywhere in the world. And essentially, I want you to don't look at the names of the devices because you're never going to remember, right? Let's start with, the next column, which is really the drug dosage. Actually, first the drug. So the first three are Sirolimus. Then the next one is Biolimus A9. There's only one balloon with Biolimus A9. And actually, there's another one, to be quite honest, Pierre. There's a Chinese one also with Biolimus A9. And then there's one more further down with Sirolimus again, which is kind of a weird balloon, but we can talk about it. But all the rest are paclitaxel, right? So majority of the balloons are paclitaxel. Keep that in mind. I'll explain to you when we talk about how a balloon is made, how a drug-coated balloon is made and how it transfers drug in a moment, why we chose paclitaxel as the first drug-coated balloon to do. But look at the dosages. They're fairly similar, maybe around 3 micrograms per millimeter squared. Anybody know what the dose of paclitaxel was on the Texas stent? It was 1/100th of this dose, just about. Okay, so this is like 100 times the dose we put on the Texas stent. Again, I'm just going to throw the little piece of information out there because it's going to make sense in the next slides why. Then, paclitaxel, there's only one way to make paclitaxel, right? So there's nothing fancy about how you make paclitaxel or limus. Actually, most of these drugs come from one manufacturer and all the companies buy them from one manufacturer, right? So it's nothing special about the drug. It's the same drug on all these balloons, made even probably in the same factory. Where the whole science comes is in 2 parts. It's this part called the excipient, which I'm going to talk about in a little bit. But basically, the excipient is how you attach the drug to the balloon so that the drug stays on the balloon during transit, and how you make sure that when you inflate the balloon, you get the most amount of drug being transported to the vessel wall and then later dissolving into the vessel wall, right? So it's super important. It tells this excipient is important for retention, right? We had drug-coated balloons in the early days where the excipient wasn't good, and literally you could take the drug balloon, okay, put it in the artery or at the vessel and pull it straight out and put it under a microscope, all the drug was gone, even without inflating it. It had no way of holding drug on, right? So the excipient is super important. And then we've also had issues where, and this may explain some of the data in the periphery, where because of this excipient, and you'll see it on another slide, is it actually, the way the excipient drug attaches onto the balloon, you may almost get like a flaky material that you see on the balloon. Those of you who've ever done a peripheral drug-coated balloon, you pull it out, and as you're moving it, you see flakes come off with some of these, right? And that's important for another concept, which we'll talk about in the next slide, which is something called particulate embolization. So how much of the excipient drug goes distillate embolizes. But bottom line, if you look at these, there's so many different ways you can attach a drug to a balloon, okay? But the devil's in the details as always. Knowing these excipients and how they were used to attach the drug can result in 2 of these balloons that are on here that look very similar, same dosage, okay? Made in the same way, having completely opposite effects. One being effective, the other one being no more effective than a naked balloon or a non-coated balloon. Okay. So the excipients, whenever you look at a drug-coated balloon, you want to know. You want to ask the person, okay, what's the drug? What's the drug dose? What's the excipient? Okay. How was the drug an excipient put onto the balloon? Okay, those are all important, right? The other important question is how much is actually being transferred to the vessel wall, but none of them I'm going to tell you because they don't want to scare you. I will tell you with some of these the truth. Go to the next slide. So let's just talk about how this sort of drug excipient thing works. So these are just normal balloons. Think of any coronary balloon and semi-compliant balloon you use every day. It's the same balloon, right? There's nothing fancy about it most times. Then the balloon is inflated on the bench, and then they coat the balloon with the drug excipient combination. And I don't know if we're going to show it later, are we? No.
We have a few slides,
A
yes.
few slides, and you'll see a little bit later. There's different ways you can do it. You can get they have like almost think of a spray can. They like spray it on and rotate the balloon as you spray it on. Others use pipettes, and like a pipette drops over the whole content of the balloon, the drug and excipient, like slowly, and then the balloon rotates, and you keep dropping drug on. Okay, you just mute whoever that is. Sorry, yeah, no, stay there, stay
Okay.
where you were, stay where you were. It's fine. We'll go to that, right? So there's different ways of doing that. Then also, once you have the whole concept is that you want to make sure the entire balloon's covered so that the amount of drug on it is the same and it's homogeneous. The great thing about a drug-coated balloon is that if you use it properly and you inflate it, you get homogeneous drug transferred to the vessel wall, right? With a stent, you don't, remember, right? Because with a stent has holes, has cells. So you only get drug where the struts are. Where the cells are, there's no drug there actually being given to the vessel wall. But because with a stent, the drug remains for longer because of the polymer or because of the way it's been attached, you get drug dosages that stay there longer, but it's not uniform. A drug-coated balloon gives you much more uniform drug distribution because the entire balloon is coated and the entire balloon will then give the drug, okay? But because of this material that's on here, okay, you can imagine, until you get it to the vessel wall, you need to protect it because the drug recipient will come off. You then get your lesion, you inflate it, and when you inflate it, the time of inflation is important because you need to give time for this mixture of drug and excipient, okay, to be transferred to the vessel wall and also to have some time to move and through the vessel wall by passive osmosis usually. Next slide. So when we think of DCBs, I really want you to think of, like, I wanted to scratch under the surface today and talk a little bit about the science as well as DCBs. So when we get the balloon then, and we've done all this stuff on the bench, there's different things that we talk about, right? We talk about the coating transfer, so how much of that's transferred to the vessel wall. And in animals, you actually see this. You inflate a balloon and you take it out and you cut, if you look, cut open the artery, okay, you see that this is what it looks like. You see the coating transferred to the vessel wall. Then you look at drug dissolution. So the first step is you inflate it, you transfer the coating. The coating has the drug, right? You now have your coating matrix on the vessel wall, which is this mixture of excipient and anti-proliferative drug. The next step is then the drug then has to dissolve, right? And dissolve into the vessel wall, and also some of it's going to dissolve into the blood and be lost, right? And then as it dissolves out of this matrix excipient mixture, it then starts being absorbed. And usually the absorption is sort of passive osmosis. It goes from high dose to low dose, right? So here's it going through endothelium neointima, and you're hoping that it's going to distribute all along the vessel wall as far as the tunica media and adventitia, right? Now, the reason we used paclitaxel in the beginning is because paclitaxel is lipophilic. And so paclitaxel loves vessel walls. So when I can literally just take a brush and go throw it on the vessel wall like that. And I'll get paclitaxel that will be absorbed. If I go 3 days later or 4 days later and did histology, I would find paclitaxel there. It's just so lipophilic that we didn't have to do very much when we started putting on a balloon. We just had to find something that would just hold it there. And that's why the first ever excipient is something we use every single day in the cath lab. It's contrast media. All right, that's how they made the first drug-coated balloon. They put paclitaxel with contrast media and they attached it to a balloon. And it actually works as a great excipient to attach your drug to the vessel wall. Next slide. Okay, so the excipient, again, there's lots of different excipients we can use. The ones that are most used are iopramide, which is, like I said, contrast media. Citrates are used quite often, okay? Citrates you'd find on the agent balloon. You'd find it on, trying to think, there's a balloon from Biotronic or something in the, can't remember the name. There's another balloon in Europe also that a lot of balloons use citrate. These are paclitaxel balloons, by the way. Urea is on the Prevail balloon. Shellac, I'm not a big fan of shellac. We used to use it before on balloons. It doesn't work very well. Shellac's the same material. If you take a tablet, you always all take tablets, the coating on the tablet, that's what shellac is. It's used to coat tablets. And then you have your drug. And interesting thing about the drug as well is you can attach paclitaxel to a balloon in different ways. You can put it on in a crystalline formation or an amorphous formation. And now we've started playing with nanospheres as well, which we'll talk about later. But whether it's crystalline or amorphous also has a very big impact. And I think we may have a slide for that. And then generally, we could put in low or high dose. We usually like high dose. 3 micrograms per millimeter squared of the balloon is the usual dose. And part of why we like high dose is a couple of reasons. Probably the most important reason, and I'll say this to you every time you use a drug-coated balloon. Just think about it. You're holding the drug-coated balloon, right, in front of you, and it's on the wire. And you go, okay, I'm going to get it to the artery as quick as possible. And no matter how slick or good you are, from the time you open your copilot, you go in, you push, you get it to the lesion without any difficulty, and you inflate the balloon, you've already lost 80% of the drug. Okay, 80, not 18, 80% of the drug. So you're only left with 20% of the drug on the balloon that's actually going to be used to be transfused and deposited on the vessel wall, and that's why we increase the dose. Okay. The other reason we increase the dose is because we know the drug is not going to stay there for 6 months. It's probably in most of these balloons, if you look at the PK studies, you can see drug anywhere from 2 weeks to a month. There's 1 or 2 balloons, you can even see it at 2 or 3 months, but it's usually, we say, about a month, and then all the drug's gone. And that's why we want that higher dose, okay? So we can actually keep drug there, and as it's being used and dissolved and absorbed, we have a dose that will hopefully last 30 days. There is some concern, though, that using that very high dose of drug may be toxic to vessel walls. And that maybe when we see, and you'll see some of the data that Pierre shows you, is that you get positive remodeling with paclitaxel more than limus. And some of our thoughts is that paclitaxel is cytotoxic. And because of that high dose, maybe that's why you get some of this positive remodeling, right? Next slide. So again, I'm going to go through this a couple of times because no one talks about this. And at the end of it, you're probably not going to remember a lot of it, but I want you, there's one line I want I'm hoping all of this is going to get you to remember. So if someone could just mute, where's Michael? He unmutes himself and he doesn't talk to me. Why she would talk to me? So the excipients is the magic part of the balloon, right? It's what's used in the coatings to coat the antiproliferative drug. They play a central role in how the drug-coated balloon works. They're responsible for drug retention during transit. They're responsible for drug to tissue uptake, how efficient and homogeneous it is. They're responsible for drug transfer and tissue uptake time, as well as drug distribution retention, and they minimize something called particulate release. So one of our concerns with drug-coated balloons, okay, is that all that material, that matrix, okay, when you inflate, if it doesn't stay to the vessel wall, and when you deflate, if pieces of that, think of like a flaky powder, now embolize down into the distal vessel bed, is that good or bad for patients? I think I can't believe anything embolizing is good. I think it's always bad. but it's something to think about, this particulate embolization. We can talk about that later. I think it may explain some of the data and the increase in some of the peripheral studies, the increase amputation rate in some of the clinical studies with drug-coated balloons. Next slide. So here again, just showing you a little bit about the coating, how it's done, right? One of the classical ways is just this micro pipetting technique where the balloon is just rotated and you just like pipette drop drug on, okay? And you can see there are 4 different balloons who use that, right? Then there's the spray coating technique, okay, where you just spray it, okay? That's sequin please, probably the most commonly used balloon in the world, okay? Then there's the dip coating technique, which is what Prevail uses, where you just take it, you dip it into a balloon. But for most of the companies, they'd like you to believe, I think to some degree it's true, is that after they've coated the balloon with the drug, is they have very special ways of folding the balloon. And by folding it, they're hoping they're going to hide away some of the drug from the vessel wall. Okay, next slide. And again, I wanted to show you just the difference between crystalline and amorphous. Just one second. So there's 2 different types of drug. This is really related to paclitaxel, okay? It can be crystalline or it can be amorphous, okay? Crystalline, think about it like, you take a bucket of ice and you crush it, okay, with like your hand, whatever, and you'd still see like these little ice particles, right? That's what crystalline paclitaxel looks like. And this is what it looks like if you looked at it under electron microscope. It results in larger particles on the vessel surface, okay? It's more difficult to dissolve though. Amorphous is much smoother, so think of like almost cotton candy, okay? Is what it looks like. It has much smaller particles on the vessel surface that are easy to resolve. Next. We should have a slider. And here again, just showing, and just think about this again when you look at that balloon, because with some of these balloons, you can't see anything, right? Especially the Sirolimus coated balloons. You look at them, they just look at a normal balloon, they don't even look white. So like, you even wonder, is there something on it? But if you were to put an electron microscope, this is what it looks like. This is what the drug on it looks like based on what drug and how on this excipient. How important this excipient is, how you use that excipient to attach the drug. I mean, look at like this is the one here on the bottom right with nano encapsulation, right? Where you're using nanoparticles to encapsulate the drug and then hold the drug onto the balloon. But I love looking at this picture because it just reminds us all and of the science of drug-coated balloons and how much science actually goes into these balloons. Next slide. So again, I want to just concentrate a little bit about crystalline and versus amorphous. Okay, they have very different dissolution profiles. So crystalline, remember, ice particles broken up, right? That's what they look like. They have a slower dissolution of the drug, but because of those those particles, they stick around for longer and the drug stays there for longer. Okay, so you actually have more sustained drug transfer, and you may get better bioeffectiveness. But remember, biological effectiveness with drugs comes at the cost also of vascular toxicity, right? The same drug that prevents restenosis also can damage vessel walls. We forget about that sometimes, right? When we use immunosuppressive drugs on people's arteries, we think only of the beneficial effects. They could potentially be detrimental effects. The amorphous, so cotton candy, very smooth. It rapidly dissolves. There's less retention. It's very uniform instead of the ice crystals on the vessel wall. And the important thing is we think less of because it's so smooth and tiny, it's less likely to embolize distally in the vessel wall. So there's a big difference between crystalline and amorphous. Next. Okay. And then another thing that's really important is how is the other drug transfer mechanism. So the transfer really relies on the fact that the coating layer has to detach from the balloon surface during contact with the vessel wall, followed by then this dissipation of the excipient and subsequent uptake of the drug molecules. we've had drugs where, and I joked with you earlier, there was one we would put it in, put it out, and we looked, there was no drug on. We've had other balloons we've taken, put it in, inflated, pulled it out, all the drug was still on, okay, all the excipient, nothing had been transferred to the vessel wall, right? So they're two extremes of that. But in that first sentence, if you read it, what's paramount to that, okay, for this to work is also the length of inflation, right? Think every time, the next time you you inflate a drug-coated balloon, I always tell you guys, leave it up for 2 minutes if the patient's tolerating. It seems boring, right? Maybe like in your mind, think about all these biological processes that have to happen in that 30, 60 seconds or 2 minutes that you're keeping the balloon inflated. The reason we're keeping it inflated longer, sure, we like to have a pulba effect of the balloon because pulba has a great effect on the balloon the longer you inflated it, right? But also all the stuff has to happen. All these biological processes have to happen. The coating layer has to detach. The excipient has to dissipate. The drug has to start dissolving into the intima and maybe beyond, so that when you deflate the balloon, blood just doesn't wash it away, right? So the next time you're inflating a balloon, you're getting bored, and you're leaving it up for already a minute, and the patient's stable, and you go, oh, should I leave it longer? Should I leave it? No, leave it longer. There's so much that has to happen in that time that's important. Next slide. I think I've bored maybe a lot of people with my passion for this whole drug-coated balloon scientific part, but just one last part again about particulates, okay? Because I think that's really important. It's something the FDA is very concerned about. When people ask me, like, Zim, how come you've had drug-coated balloons since, over 10 years, almost 14 years in Europe, okay, and you were using them, and it's taken such a long time to get drug-coated balloons in the US? This picture on the right is the main reason, actually. Okay. The FDA has been very concerned about particulate embolization. So what does this mean? The FDA is very concerned that we put the balloon in, we inflate the balloon and deflate the balloon. Sure, some drug has gone to the vessel wall, but they're concerned most of it is actually embolized distally, drug and excipient. And they want to know what's happened to that drug and excipient, right? So when in the animal studies we've had to do to develop many of these drug-coated balloons, we had to really, after you've inflated the balloon, you sacrifice the animal. You then have to provide the FDA. You have to send to histology, but you have to provide FDA the histology of the heart, all the arterial beds that were perfused, the kidney, the lungs, the liver. And in all those histological specimens, you have to look for the drug and the particular material. Because remember, some of these particular materials we use are not naturally occurring materials, right? And what's interesting, and I can't mention the name of the balloons, some balloons, when they did that, they found there was stuff like this everywhere, all around the animal's body, right? There were little infarcts even caused by these particular materials going everywhere, okay? And even with a balloon, some of you may know, and I don't know if we have that slide, there's animal data showing that in the distal area of the vascular blade, you may see particulate material, right? So I'm a little concerned about particulate material. It's more common with certain types of drug-coated balloons versus others. In general, it's more common with paclitaxel than sirolimus. But even in paclitaxel, it's probably more common with the crystalline formations that have those bigger particles, okay? Have I seen any negative consequences? So in my career, I've seen a couple of cases where I've done long diffused LAD lesions, and I've done 2 to 3 30 to 40 millimeter drug-coated balloons of a long vessel, and I've seen slow flow afterwards. Okay. Now, was that slow flow maybe from embolizing plaque or other materials? It's possible, but I also think it was potentially caused by particulate embolization. I mean, I had one so bad that the patient ended up on mechanical support. So it could be really bad. So again, these are all things we need to have in our mind and think about when we use these technologies. Next slide. Why don't you carry on from here?
Yeah, no, So this is just a few more slides, just on the concept that we reiterated in the past minutes. So the drug is in a balloon, needs to leave the balloon, and then needs to go on the vessel. And these are just some details that I don't think we need to stop by on the how the balloon is made itself, but the concept is what we talked about. And I think different excipients really allow us to have, especially with sirolimus, but also with paclitaxel, to have the drug transfer from one place to the other. We need to think that the stent is covered and it stays there. So there's really, as long as the drug is on the stent, it's going to be on the vessel wall as well, because you're going to have in contact with the vessel. Well, this is not the case with drug with a balloon. So this is why we're spending so much time describing these details. And in terms of downstream particulates, this is just some images that really make you understand what's happening, what might be happening. And high coating crystallinity and hydrophilicity, as we said, really might produce more particulates than others. And this is a recent study, was presented at CRT 2023 and was published just 1 month ago from the lab of Alok Fin. And it really shows you what Dr. D was saying just a couple minutes ago, that some balloons do yield more often downstream emboli than others. And some balloons do yield more myocyte necrosis or scarring when they sacrifice the pigs and we look at histology. These are just a few images, okay? And this is just some inserts from the study where Alok Fin really says, well, maybe this might be also due to the size of this emboli. And some balloons might have larger sizes, other balloons might have smaller sizes. We're talking about microns here. So some balloons have single-digit micron particulates. Other balloons have about 50 micron particulates. So it's really different when you see in the magnitude of the microns that we're all talking about. And we've spent all the time with the excipient part. I think it's reasonable to spend a little bit of time on the drug part. So sirolimus versus paclitaxel. In terms of all the properties that we discussed, paclitaxel in general might be considered more favorable in terms of his transfer from the balloon to the vessel wall, thanks to the lipophilicity. But as we said, sirolimus is not cytotoxic and has a wider therapeutic range. So it really, nothing comes for free, as usual in life. And I think we need to really focus on the difference between these balloons, both in terms of pharmacological details, but also in terms of clinical outcomes. And this is just one balloon with sirolimus. This is one of the newest balloons. It's called the solution balloon. So this balloon is really coated with the same bioresorbable polymer that was the bioresorbable polymer used for bioresorbable scaffolds. So it's PLLA. And these nanospheres with PLLA, they really hold sirolimus, and they allow the transfer of sirolimus from the balloon all the way into the vessel. And you can really see that, preliminary data shows that up until 90 days, the drug concentration is superimposable to that of a Xyant stent. So companies are really trying to do the best to have a drug release kinetic that is as close as possible to that of drug-eluting stents without a strength in place. This is another sirolimus balloon. This is the Magic Touch from Concept Medical, developed in India. And we're talking about how the balloon is folded. And in terms of excipient, the drug here is housed within a 20 to 200 nanometer sphere of hydrophilic head coating and lipophilic tail, so that the drug is lipophilically coated and can be transferred to the vessel wall. And when we look at data from clinical studies,
and then just go back. So,
yeah,
I mean, we've been trying for years to get Limus onto a drug-coated balloon, okay? And it was just impossible. We didn't have the technology. And really what's changed it is the fact of being able to hide because the problem with sirolimus is, everybody, when we were in the ages of, there were just two drug-eluting stents, Cypher and Texas, and then Zynes came along, Cypher and Texas disappeared. But even before Texas disappeared, we knew Texas was very potent, but because it was so potent, we probably saw things like more very late stent thrombosis, and we saw more things like aneurysm formation after Texas. And so in Europe and the US, there was like no more Texas stents. They do still exist in some countries. I think in India, there's still Texas eluting stents, and then there may be some in China too, but they really disappeared. And when that happened, we tried already to try and make Limus-coated balloons. It was extremely difficult, and we really didn't have the technology. It's only in the last few years where we now have the technology available to create these microparticles. And the whole idea with the microparticle is Limus is so hydrophilic that if you just put it in a balloon, it just washes away. It loves being in water. So you're trying to hide away the drug from liquid, from blood, and you use these lipoparticles. And as you know, anything that's lipophilic attaches to the vessel wall and is absorbed to the vessel wall. So that's the whole concept, is that you use different types of nanoparticles to hide the drug away so that you can then attach it. And then you can do all sorts of cool things with biochemistry on those nanoparticles to how long they will stay in the vessel wall. You can like really fine tune this stuff that you can make it stay 8 days, 28 days, 58 days. You can really play with that. And the reason that's fun is because we actually now have a new balloon we've been working on for the last few years where using nanoparticles, we can put paclitaxel and sirolimus into nanoparticles on the same balloon, right, in different dosages, so that we can hopefully get the beneficial effects of both, right? We can have maybe a little bit more sirolimus, because sirolimus is cytotoxic, so it doesn't damage the vessel wall, but keep enough paclitaxel on our balloon so that we can get some of the positive remodeling that you see with paclitaxel balloons. And the other thing I like about nanoparticles is that the risk of distal embolization or particular embolization is zero. These things, even if they're embolized because they're nanoparticles, they don't cause microvascular obstruction.
Yes, and in terms of clinical data, I think out there right now there's growing amount of evidence, and on this slide you can really see in the last year there's been 2 meta-analyses that have compared outcomes of patients treated with paclitaxel or sirolimus-coated balloons. And on the left-hand side was sirolimus versus paclitaxel, and the other one was Limus DCB, so included also Limus versus paclitaxel. And in general, the results are pretty much superimposable, meaning that we see in clinical practice, so when we do follow up angiographically, that paclitaxel does result in a larger minimal lumen diameter, larger area, and in general, larger vessels that follow up at 6 to 9 months. And the sirolimus still clinically superimposable, but in terms of angiographic outcomes, it does, just merely measuring the diameter of a vessel at follow-up is inferior to paclitaxel. And in clinical outcomes, we have seen no difference. So whether these differences in angiographic outcomes do transfer and actually apply to clinical outcomes, so far there's no evidence. Is it just merely an issue of number of patients enrolled? We need to see. This study that we presented was more than 1,500 patients, and we really saw no difference between these patients. And in terms of angiographic, this is one of the studies that have shown this the best, is the Transform 1 study, compares sirolimus Magic Touch versus paclitaxel Sequin Please Neo, and it really showed that Magic Touch was not inferior in 6 months angiographic net lumen gain when compared to the paclitaxel balloon. And those were de novo lesions, and in this last Siblent ISR randomized trial, they actually shown that in ISR, actually, the 2 technologies are comparable, even though if we look at the fine details, we still might see that there might be some kind of advantage, even if not statistically significant, in favor of paclitaxel in terms of pure angiographic outcomes. So I think here we get to the point of how we use DCB.
I think we should stop here.
Yeah.
Okay. I think let's leave some time for questions. And then maybe we'll do part 2 next week. And in part 2, we'll talk about how do we actually use DCBs. We'll go through some of the indications, ISR, small vessels. We'll talk about technique. We'll be a lot more practical. But I think I'd rather not rush through it. If everybody on the call agrees, let's split into two. I wanted to leave time for questions. I see while people are warming up, I see there is one question, and it's a question from Madai. Go ahead, Madai.
Yeah, I'm wondering, the other drug that is used in stents is everolimus. Why is not a drug that is used on DCBs? I'm assuming there must be an issue with it.
Yeah, so the whole reason it's all about patents and licensing. So I think Novartis made everolimus and they licensed it, and Abbott has the license. But all the everolimus is made in one place, and both Abbott and Boston Scientific buy their everolimus from one factory. Okay. So it's just like, if you're a startup company, you cannot go out and buy everolimus to put it on a balloon. You can go out and buy paclitaxel easily. You can go and buy Sirolimus because it's been around for so long, the patents are all gone. And even Biolimus A9, I think, is owned by Biosensors, the patent, so you have to pay them and then give it. But, for years, Boston was paying Abbott, I think, for stents, right? And when the first Synergy and Zyme stents came out, I don't know if you guys know this. When the first Synergy and Zyme stents came out, both stents were made in exactly the same factory. And then after they were made, they would split in the factory up, and one would get boxed in a Abbott box, and the other one in a Boston Scientific box. It used to be exactly the same stent for about the first 5 years they came out. It was then made in the same factory. Okay, but it was sold by a different company and a different box. It's not the true anymore because Zyme still owns their durable fluoropolymer as a way of attaching drug, whereas Synergy, to be able to separate from their production line, not have to pay Abbott for their stents, they then went to a biodegradable polymer. And then eventually they also changed they changed their stent platform, okay, to Synergy. Actually, there was a stent before. I don't know if Michael remembers what it was called. There was a stent before Synergy with Boston that was identical to Zyme's. But then when they changed Synergy, they changed their stent platform. And they had lots of issues with that platform, right? Because the Synergy stent was the most common stent that had longitudinal stent deformation. And so they've had to change that platform more than once. But it's just interesting if you go back and you look at the history books, right? And that's what I started putting in drug-eluting stents. I remember it was like the funniest thing. You'd have these two boxes, and you knew that one looked one color and said Boston, the other one looked another color and said Abbott, but they were made in the same factory. So it's just a question of drug licensing, really. That's why everolimus hasn't been used.
Yeah.
Long answer to a simple question. Sorry. Any other questions, guys?
So does the
Hi, go ahead.
Okay. Go ahead, Sharon.
Sorry, I was just wondering, does doing arthrectomy beforehand or whether the vessel is calcific, fibrotic, does that influence how the drug attaches to the vessel?
We'll talk a little bit about that more next week, all right? Because I think there is some data there, but I'll just say this because I don't want you to have to wait till next week. So one of the areas of greatest discussion for all of us is, do drug-coated balloons work in calcified lesions? Okay. There's really no clinical data. The benchtop data is confusing as well, because if you just have, say, a 360-degree calcified lesion and you just put drug on it, I mean, that calcified lesion is a rock. Drug's not going to do anything, right? So even if you do just atherectomy and you shave, I'm not sure you have an impact. You literally have to cause fractures into the calcium. So there's some data on the bench that, because also there's no bench models, animal models of calcified coronary lesions, right? So we can't study this on the bench because there is no models, right, in animals. I wish I could find an animal with calcified coronary arteries. It would be great. It'll be a lot of fun. But what we think, and there's maybe some suggestion of it, is if you have a calcified lesion and you crack it, and I'm talking about like a 360 degrees crack, and you crack it and fracture in multiple areas where you're exposing blood to the media, okay, and to the living cells that are below the calcified cells, then yes, they could be impactful. And that's why if you're going to treat de novo or even calcified neoatherosclerosis with a drug-coated balloon, you really have to not just shave it, you have to create breaks in the calcium. You have to fracture the calcium to be able to allow the drug to get to behind the calcium.
Thank you.
Louis,
Yeah,
you had a question, and Sahul. So I'll go Louis first and then Sahul.
yeah, I have a question. Just before asking my question, we did a case for a superior patient, Andre and I, a few months ago, and supercalcified vessel, rotational atherectomy, IVL, and we treated only with DCB. I don't know if you remember this guy, but the follow-up angiogram, it looked amazing. So when the vessel is well prepared, I agree. I think it might work. My question is regarding late lumen gain with paclitaxel DCB. Do you think there is a value in, because the drug is essentially gone after a month, maybe a bit more in some instances. Do you think there's a value in going back to the cath lab and repeating paclitaxel DCB angioplasty to get more of that late lumen gain, or not?
It's a good question, right? I guess I'm a little bit concerned if you were to go treat the artery too soon afterwards, about the negative consequences. So just to take a step back, and we'll show you some of the data next week, right? But all the de novo small vessel studies we've done with paclitaxel have shown that when you look at late lumen loss at 9 months to 12 months, what you see is you see this sort of late gain, right? So the late lumen loss can even be negative. So whenever you have minus late lumen loss, okay, so when you measure it, so remember, just for those of you, because I realize they don't teach what late lumen loss is anymore. What late lumen loss is if you take the you take the minimal, or the RVD, the MLD, sorry, so the minimal area, okay, at the end of the procedure, and then you subtract from that your MLD at follow-up, an angiographic follow-up. That's your late loss, right? So how much, if you start with an MLD, an artery, you made the MLD 2.5 millimeters, right? We expect that with any technology in the coronaries, if you go to that same area and you measure your MLD at 9 months, you expect like a late lumen loss, even with a stent of maybe probably, depending on the stent, 0.2 to 0.4 of late lumen loss. Okay, so your 2.5 will no longer be 2.5, it'll be 2.1, right? So it'll be 2.5 minus 2.1 gives you 0.4 late lumen loss. That's how you calculate late lumen loss. With drug-coated balloons, we've seen in some vessels, it's 2.5 at the end of the procedure, and then you come back, and it's 9 months, it's 2.8. Okay, it's 0.3 bigger, or it's 2.6. Okay, and so when you calculate the lumen loss, it's minus 0.3, it's minus 0.1. Okay, millimeter. That's negative late lumen loss, right? Negative late lumen loss means you're getting lumen enlargement. Okay. We've mostly seen that if you compare the data, and you saw it a little bit from Pierre, we've mostly seen that with paclitaxel. We've almost not seen that with Limus, especially the percentage of there's some late lumen loss. Sorry, there's some negative late lumen loss, but it's mostly with paclitaxel, where if you draw a distribution curve of your late lumen loss, you see it like shifted more towards the left. So more lesions will have negative late lumen loss. Now, the question is, if they both drug-coated balloons, right, and they both leaving nothing behind, there's no cage, why does paclitaxel give you minus late lumen loss and lumen enlargement, and Sirolimus doesn't, right? It has to do with the biological properties of paclitaxel, right, and the fact that paclitaxel is cytotoxic. So it's damaging the vessel in a sense, all right? I mean, you're getting late lumen loss not because you're suddenly making the vessel more compliant or more responsive to, acetylcholine or vasodilators, because if you were, then why doesn't the limus group also have it? It's probably happening because you're toxic to the vessel wall and you're actually creating mini, medial necrosis and mini aneurysms that don't look like aneurysms. That's probably what we think. And as we do more and more drug-coated balloons, and there have been some cases where people have overly aggressively treated the same area more than once, and one of the areas we worried about, Louis, and this affects you, is also about CTOs, right? Where if you've done lots of subintimal tracking and dissection, and you're now exposing more drug even to the adventitia, could paclitaxel be worse than limus? We don't know, but there's some concern because you are causing more vessel damage, and that's why you're getting more remodeling, but more excessive remodeling is called an aneurysm, right? Remember that, right? That's what excessive remodeling is. You could cause an aneurysm. So that's kind of why I'm reluctant about that a little. I'm worried about aneurysms. We published at least 2 papers, and we're doing a 3rd paper right now, looking at in patients that are very high risk for restenosis, so like diabetics, patients with end-stage renal disease, in areas where there's a very high risk of restenosis, doing drug-coated balloons and stents to the same area. Okay, so you give double drug, one's a limus, one's a Taxol, and you can now work on 2 different mechanisms of action to prevent restenosis. And the data, it's all retrospective data, looks very good, but when we showed it to the FDA to try and do a clinical study, they were like freaked out. I can tell you now, they're like, oh my God, you guys have to show us a lot more animal data to prove to us you're not going to cause aneurysms. So that's the concern, I think, from a safety point of view. Sorry, again, long answer, but I like to give you guys a little bit of the history of this area because we've been working in this area so long.
Thank you.
Saul?
Morning, Dr. Stephen, Andrea. Thank you for this talk. Thank you, Pierre, as well. It really helped us understand some of the mechanism of actions of these balloons. My question may not be simple to answer, but if you had all of the available balloons that you use in Europe, which one would be the one that you would prefer to use and why?
So I have to be, you know what? We don't have PS answer this question. I have to be politically savvy and not make any of the companies unhappy. No, I'm kidding. I have to say, any of the balloons we're using right now in clinical studies, I feel comfortable with, right? So the ones that are going to be used in clinical studies in the US are Magic Touch and Solution for Sirolimus, Prevail from Medtronic, an Agent from Boston Scientific for paclitaxel, and I think eventually Sequin Please will come as well. I think those 5 balloons, I feel very comfortable with all 5 of those balloons. There's thousands of patients who have been treated, and there's lots of data. The sad part, and that's kind of what is when you look at the clinical data that's been done in Europe, there's a lot of, and I contribute, there's a lot of retrospective studies, a lot of multi-center retrospective studies, there's a lot of prospective registries. What's missing from Europe are large-scale randomized studies, right? The largest randomized study that's important is basket small and small vessels, but like big, like the agent, we get a lot of criticism in the US about the agent study from the Europeans saying, oh, I can't believe these Americans did a study where they randomized a patient to POLBA, right? But hang on, this is the first, even though these technologies have been around for 15 years, this is the first study that actually did that in such a large scale with a clinical endpoint, not with an angiographic endpoint, right? So we're really doing the studies now that I think are going to define the field and are going to help improve guideline indications. So if I look at the next studies that are coming down the line, like Magic Touch, okay, this is the largest study for a sirolimus-coated balloon, okay? We'll actually figure out, does it work in restenosis? Because all the data prior to this is mostly single-arm study or small randomized studies with an angiographic endpoint, not with a clinical endpoint. So that's why I say to you guys, we're so lucky in Monty. We're involved in at least 3 of these clinical studies. There's a 4th one coming. we have an opportunity to really understand this field and to define it for all future interventional cardiologists who are going to be working in this field, because you only really know if something works when you test it in a well-conducted, well-powered, randomized clinical control trial with a clinical endpoint. Up until that point, it's just, it's us experts talking on meetings about what we prefer, what we like the best. It's a lot of bullshit. Come on, right? You have to do the study, right? And I give you an example of recently, the Accurate valve from Boston, right? I implanted hundreds of those valves when I was in Europe, okay? I was involved in the clinical study. It's still being implanted. And we all thought that the data was great. Then we did a randomized study that was large enough, and we went, oh, shoot, maybe there's a problem here, right? The patients who get the valve have a higher risk of underexpansion, stroke, and maybe even mortality. There were 20, 30, 40, 50,000 cases done already, and we didn't know that information, right? So we got to do the randomized studies.
Thank you.
Any other questions?
I have a question, Dr. Leo. Good morning.
Yeah.
It was a great talk. Do you see any utility of using DCBs in SVG graft, especially with the distal anastomosis?
So I think anastomotic sites, yes. The actual vein graft itself, I'm not so sure, right? Remember that histopathology, if you look at a vein graft degeneration histopathology, it's a very different type of degeneration compared to what we see with atherosclerosis, because it's accelerated arterialization of a vein, right? And you should look at some histological pictures of what a vein graft looks like when it's degenerated. It's actually pretty scary. And I have to be honest, nothing works in vein grafts. We have tried everything. We've tried bare metal stents. We've tried cover stents. We've tried drug-eluting stents. Nothing works. Even drug-eluting stents, I hate to break it to you, but in a vein graft, if you look at the data at 2 years, okay, the curves with the bare metal stent and vein graft all come together. So the drug-eluting stent helps you early on, okay, it delays the restenosis, but eventually you catch up. Right. And I think that's why, Louis gets so excited when he sees a vein graft degenerating, right? Because he knows he's going to open the native vessel, because the outcomes from doing native vessel PCI are so much better than doing vein graft PCI. Louis, I saw you coming off mute. Say something.
I agree.
But basically, if you were like, I really like, why I really like drug-coated balloons is like a Lima anastomotic site. Okay. I really like drug-coated balloons there because, if it dilates well and it looks good, number one, I can track the balloon down better. But also, I'm not jailing, the retrograde flow that's going up, retrogradely up the LAD. So I really like it for that. One of the issues I will have is, and I realized that on Friday, again, is the profile of an agent balloon is not great. I've had now a number of cases, and I've forgotten about it from when I was in Europe, because we were mostly using Prevail and Magic Touch. I've now thrown away, in the last month, I've thrown away 2 drug-coated balloons that I could just not get down an artery. I'd gone down, I'd prepped the lesion, I'd gone down with non-compliant balloons, everything. And then when I took the agent, it just would not go down. And I had to throw it away, basically. I couldn't inflate it, right? Which is a very expensive waste, a $7,000 mistake, right? So you'll see when we start doing Prevail and we do more magical touch, their profile is much better. Okay, guys, I think we'll see you next week for part 2. Get to work. I'll talk to you all soon.